Proteins are an important class of biological macromolecules present in all organisms, made up of elements such as carbon, hydrogen, nitrogen, phosphorus, oxygen, and sulfur. All proteins are polymers of amino acids. These polymers are also known as polypeptides and consist of a sequence of 20 different amino acids, also referred to as residues. To be able to perform their biological function, proteins fold into one or more specific spatial conformations driven by a number of non-covalent interactions such as hydrogen bonding, ionic interactions, van der Waals forces, and hydrophobic packing. In order to understand the functions of proteins at a molecular level, it is often necessary to determine the three-dimensional structure of proteins through complex techniques such as X-ray crystallography or NMR spectroscopy. However, proteins are not static and rigid structures. The polypeptide backbone and especially the side chains are constantly moving due to thermal motion or kinetic energy of the atoms. But many biological processes are mediated by molecular movement and dynamics. When studying the structure of a protein, different techniques give different information about the motion in macromolecules. X-ray crystallography allows a time-averaged snapshot of the structure which appears as if it were frozen. One way to understand the motion in proteins is to use computer simulations called molecular dynamics. With this technique, changes in protein conformation are calculated as time-dependent behavior of a molecular system. Molecular dynamic simulations have provided detailed information on the fluctuations and conformational changes of proteins and nucleic acids. These methods are now routinely used to investigate the structure, dynamics, and thermodynamics of biological molecules and their complexes. They are also used to explore the macroscopic properties of a system through microscopic simulations, for example, to calculate changes in the binding free energy of a particular drug candidate or to examine the energetics and mechanisms of a conformational change. In this exercise, we will use Discovery Studio to examine a protein-ligand complex. Dr. Holger and Gerhard Kleeb wrote a recent review concerning the study of protein-ligand complexes and identified several important considerations such as electrostatics, solvation, desolvation, and conformational changes in the ligand and receptor. We are now going to explore these contributions to the interaction of a ligand with the receptor. The example we show today here is a vitamin D receptor which is responsible for the expression of a variety of genes involved in calcium metabolism, bone formation, and cell growth and differentiation. Understanding vitamin D receptor conformational changes resulting from interactions with bound ligands may help to identify and treat persons at risk for disorders such as osteoporosis, breast cancer, and prostate cancer. The vitamin D receptor domain with the bound ligand is now displayed. The three-dimensional structure for vitamin D receptor is obtained through X-ray crystallography. The ligand is shown in yellow. The structure has been pre-configured with force field parameters required to calculate the molecular dynamics. We will only run dynamics on those residues in immediate contact with the ligand and the ligand itself. Thus, we will restrain amino acid residues beyond a certain distance from the ligand. To choose those resistance, we use a simple distance criterion around the ligand. The highlighted ligand is in the middle of the protein, and as we select the amino acid residues surrounding the selected ligand, you will see most of the protein residues selected. If you look closely, you will find that the residues in contact with the ligand are not selected. If the center of mass of any of the residue is within 7 Armstrong units of the ligand, which is the distance criteria defined, then the entire residue is not selected. This whole operation selected all the residues outside those in contact with the ligand, and the small blue cages surround the fixed atoms. The rest of the protein is flexible to move under this molecular dynamics simulation program. All the parameters required to perform this experiment have been made, and the experiment itself might take less than a minute approximately. After the simulation process is complete, we may view the animation of only the residues in contact with the ligand. The structure in the new 3D window will begin to move. There are only 10 frames in this animation, and once the animation frames complete, it will resume again. From this molecular dynamics simulation, it is possible to plot the energy. We can now plot the potential energy of the system by creating a plot and choosing x and y axis as steps and potential energy respectively. From this plot of potential energy over frames or steps, it is possible to identify the lowest energy frame of the trajectory. The graph that we thus obtained here is interactive, and each point can be interactively mapped on to the conformations generated by this molecular dynamics program. Our interest is particularly of a conformation where the potential energy of the system is very low. We may also calculate the interaction energy from the displayed trajectory. We may calculate the interaction energy between any set of atoms within the molecular system. In this case, we can calculate the interaction energy between the protein and the ligand. In this example, we are selecting the entire receptor protein and the entire ligand. We could have chosen an arbitrary set of atoms to define the two groups. For example, we could have chosen a single amino acid residue and calculated the interaction energy with the ligand. However, this job will calculate the interaction energy between the ligand and the protein receptor as a whole. The run will complete in less than 1 minute. This experiment has examined each frame of the trajectory and calculated the interaction energy for each frame. The data is represented in columns such as interaction energy, van der Waals interaction energy, electrostatic interaction, and the frames. This exercise demonstrated one approach to use molecular dynamics to examine protein-ligand interactions in Discovery Studio. The starting structure in this example was derived from an experimental X-ray crystal structure, but could have easily been a more hypothetical structure, for example, a docked ligand to a homology model of a receptor. Once we had a starting structure, an energy minimization was performed following a short molecular dynamics simulation. It could be used to examine the binding site flexibility or the induced fit of a protein and ligand. The simulation was in vacuo, keeping the exercise simple. In much complex exercises, we could have seen how to include various solvent models in the simulation. After the dynamics, we calculated the interaction energy between the ligand and the protein receptor. The method to calculate the interaction energy used here is somewhat simplistic, and more sophisticated methods are available.